I'm going to start to record now. Hello, everybody, to be here today. It's nice to be here again in this opportunity to present to Federico Roda. Federico is a biologist from the National University of Colombia with a PhD in evolutionary biology from the University of Queensland in Australia. Just after finishing his PhD, Federico moved to Cambridge in Massachusetts as a postdoctoral fellow and research associated to the Dr. Robin Hawkins lab at the Harvard University. In 2018, Federico moved to Colombia to lead the mass plant tandem group at the National University of Colombia. From there, Federico studied the evolution of plants and their adaptation to environment, but integrate genomics with physiology and ecology. In the past, he has researched about pest resistance, adaptation to coastal habitats, and the evolution of reproductive systems. Today, he's going to tell us about how omics uncover the mysteries of the Angel Trumpets, the genus Brugmansia. Thank you, Federico, one more time to accept this invitation. Can please share your screen and start with your presentation. And thank you to all to be here.
Okay, thanks, Andres. I hope you can hear me. I'm going to share my screen now. So let me know if you can see it. Is it fine?
It's fine, yeah. I can see you.
Okay, great. So hello, everyone. So my name is Federico Roda, and today I'm going to talk about the mysteries of Angel Trumpets and how omics can help solving some of these mysteries. This is part of the research that we do in the lab that I lead, which is the Max Planck Tandem Group in Evolutionary Genomics of Specialized Metabolism. Is quite a mouthful, but what we try to do in my lab is to use an evolutionary framework to understand how plants produce medicinal substances. And for that, what we do is to integrate phylogenetics with genomics and metabolomics. My lab exists since 2018, and it has been growing steadily. We are now composed of 6 people, although some other people are entering. And it's a really multidisciplinary and collaborative team. And we work in multiple areas. For example, Gina and Pablo work in the evolution of acyl sugars and steroidal glycoalkaloids in solanum. Luis Miguel works in the evolution of anthocyanins in potato. And David and Sofia lead the work on Brugmansia, which I'm going to show you today. But we really work as a team, so everybody has collaborated to the work that I'm showing you today. Max Planck Tandem Groups have been created to create research links between a Colombian institution, in this case, Universidad Nacional de Colombia, and Max Planck Institutes in Germany. In this case, we collaborate with the Max Planck Institute of Molecular Plant Physiology in Potsdam. And the idea is that for my research, it is necessary to establish these very close collaborations with people in the Universidad Nacional. Like, for example, I work with Dr. Clarines Orozco and Dr. Clarines Bermudez in this research of Brugmansia. And in the Max Planck Institute, we work with Alice David Fermi, Federico Escosa, and Saleh Axcle, among other people. We also are collaborating in this Brugmansia project with Dr. Borno Sadel in RWTH University. So today I'm going to talk to you about these beautiful plants belonging to the Brugmansia genus. And as you might have realized in my biography, I am new in the field of Solanaceae. I have been fascinated by these plants all my life, but I'm new in the field. So I wouldn't really want to get into arguments with people that have worked for a long time in these plants. However, and these seminars have been really, for me, an amazing opportunity to learn a lot of things about the Solanaceae. One thing that I took great aim in the previous talk was, I remember Leonie Moll mentioned that the Solanaceae was a very dramatic family, and I do agree with that. And she was trying to find adjectives to describe, and she said that it was not particularly charismatic. And I think that is not true, at least for these plants, as you can see in this image. And there are a lot of other adjectives that we can assign to the Solanaceae and to the Brugmansia in specific that I hope you will agree with me at the end of this talk. So this talk will be divided in 3 main sections. I hope I will have time for the 3. Otherwise, I will skip the last one. First, I will talk about the history of the plants, the metabolic variation, and finally, I will talk about leaf variation in the group. So as an evolutionary biologist, I have always been interested in the close relationship between plants and humans and how this interaction can shape the biology and genome of the plants. And the Solanaceae is really a great family to study this because, as you all know, it contains crops like potato. And this has been essential during the history of humankind. For example, they made possible the growth of the Inca Empire in South America. And they were later transported to Europe and made possible also a huge growing population in the 19th century. But the population of Europe became so dependent on this crop that when the late blight attacked this crop, it caused a great famine that sent migrants around the world and shaped the ethnography and the history of the world. This continues to be the case in Colombia right now. We are in a huge crisis with potato farmers. And genomics have recently allowed us really to get an insight on this interaction between humans and plants and understand how humans shape the genomes of these plants, shape their biology. For example, this recent study in potatoes showed the relationship between cultivated potatoes and their wild relatives. It also showed regions of the genomes that were selected that were affected by domestication. Another great thing about genomics and its power to help us understand this interaction between humans and plants is that genomics can help us understand the past when we look either at archaeological samples or at samples collected in herbariums. For example, these 2 studies in potato have used herbarium samples to understand how the potatoes adapted to the European weathers and also how Phytophthora, this pest that caused the Irish famine, caused this devastation. However, we humans don't use all the plants as crops. We also use them as narcotics and medicines. And here I bring up this example of a substance that is produced by plants, cocaine, which has had very, very powerful effects on the economy, arts, geopolitics, and violence in the world. And these effects are very complex and very strong. And nowhere in the world knows that better than my country, Colombia. However, because these interactions with plants like erythroxylum, which is the plants that are used to produce coca, has been very long in South America, we can again use genomics to understand how this history happened, how the coca was domesticated, and how this domestication shaped the genome of these plants. And this is, for example, an example of a very recent study where they use herbarium samples to understand the domestication of coca. So cocaine is not produced by solanaceous plants. However, it belongs to a family of secondary metabolites known as Tropane Alkaloids that are produced by solanaceous plants. And these are 3 examples of the most important Tropane alkaloids produced by solanaceous plants. And these are substances that have a lot of medicinal uses, but they are very potent hallucinogenic and narcotic. And because of these effects, they have been very important in the history of humankind and the Solanaceae, and this interaction between humankind and the Solanaceae. And because of the medicinal properties of these substances, a lot of the plants that are used, for example, in this case, in the old continent for traditional medicine and witchcraft, are solanaceous plants that contain tropane alkaloids, as shown by all of these names that I put here. All of these are solanaceous plants that contain tropane alkaloids. And this is true not only for Europe, but across all continents. Solanaceous plants that contain Tropane Alkaloids have been really important in medicine, but also they have impacted all the culture. And I could spend a long time talking about the European Tropane alkaloid producing plants and their impact in culture, but today I'm here to talk about the Brugmansia, which is a neotropical South American plant that was so beautiful and mysterious that even today it has an impact in the art. Well, this is not so recent, it's like 2 centuries ago, but this is a drawing from Alphonse Moucha that shows some Brugmansia plants. We can also see it in, for example, this recent, from the last century, drawing from Alfredo Ramos Martinez. But Brugmansia is a plant that has been used by people in South America for a very, very long time. And because there is no archaeological evidence of its use, we have to rely on the art to know about its use. For example, some of the oldest references to the use of these plants come from the Obelisco Tello de Chavinahuantar in Peru that seems to depict some fruits of Brugmansia. We also can see depictions, possible depictions of Brugmansia plants in very old Quimbaya spin whorls, as well as in art from the Incan Empire. However, because the ancient art is very abstract and because there is no written record of the uses and importance of these plants in ancient pre-Columbian cultures, we have to rely in more recent evidence to know the importance of these plants in indigenous culture. So here I'm showing a Guambiano drawing that is, I think, from the beginning of the 20th century, that is accompanied with a text that shows how important and how revered and respected these plants were for the Guambianos, and they were associated in general with evil. Even if they were used a lot, they were associated with evil and people were scared of them. And there are a lot of anecdotes of the use of these plants by indigenous people. One that is documented by Humboldt and comes from the conquistador Gonzalo Jiménez de Quesada, who is the founder of Bogotá, where I am now. And so when he arrived with his troops to the savannah of Bogotá, he was given a beverage containing Brugmansia seeds. And he got intoxicated with all of his troops. Very, very intoxicated, but they recovered from that intoxication. And here, what I'm showing here in the text is what he wrote after the intoxication. So that shows how strong these hallucinogenic plants can be and how can they change sometimes the minds of the people. Unfortunately, the effect of the changing mind was not too long. So for the conquistador. Because during the conquista of America, the conquistadores were not particularly interested in describing the botany or the ethnobotany of South America. Maybe our first references of the botany and uses of these plants come from the old suspects, the botanists, naturalists, expeditionaries from the 18th and 19th centuries that you are probably all familiar with. However, the golden age of the study of Brugmansia really happened between the 1930s and the 1960s. And it was motivated by people in the Universidad Nacional de Colombia and Harvard University, Hernando García Barriga in the Universidad Nacional, and in Harvard University, Richard Ewald Schultz, and some of his students like Tommy Lockwood and Melvin Bristol, which I'm not showing here. And in part, this was a golden age because of the great researchers and expeditionaries that these people were, but also in great extent it was because they work with indigenous people that gave to them their knowledge and helped them understand these plants. So they were good enough to get the indigenous people giving them their knowledge, and also good enough to hear what they were telling them. And so if you don't know, Richard Schultz produced a lot of academic and scientific literature, but he also produced some more divulgatory books that come to become like some hippie cult books, like in this case, The Plants of Gods. Some of the students of Schultes, like for example, Wade Davis, wrote some books that also kind of make more popular the work of Schultz in South America. And One River is one of those books. Also, this book inspired a film like El Abrazo de la Serpiente, who made popular their work with indigenous people. During the last part of the 20th century, there was kind of a decay in the interest in studying Brugmansia. Unfortunately, after the 2000, there was again interest in studying these plants, and I bring this book, Juan Duch, to kind of as an example of that renewed interest. This is a book that is really a fantastic compendium of all the information in Brugmansia, and has some photos that really make you fall in love with these plants and with their diversity. So coming to the biology of Brugmansia. Brugmansia is a relatively small genus. It is composed of between 5 and 7 species. I say that because some classifications don't accept Brugmansia insignis and Brugmansia vulcanicula as species. But these are the 7 species, or 5, that we have in the clade. Most of what we know about the molecular phylogenetics of these plants comes from this research by Dupin and Smith, where they used 3 nuclear genes to do the phylogenetics of this group. And what they found was that Brugmansia is actually a monophyletic group that is sister to Datura. Datura, as you might remember from some of my previous slides, is also a Tropane Alkaloid containing plant that is used a lot by traditional medicine, both in Europe, in the old continent and the new continent. So Brugmansia was believed to be part of Datura for a long time, and it changed between genus status and making part of Datura several times. So this shows that they are sister groups, and also that Brugmansia is divided in 2 clades: the Brugmansia clade, shown here in blue, and the Spherocarpum clade, shown in red. So these species from these 2 clades can not hybridize between them, but they hybridize frequently within species of the clade. And the 2 clades are mostly distinguished by their fruit shapes. So it's more elongated in the Brugmansia clade and more ovoid in the Spherocarpum clade. Brugmansia is a South American genus, and with the exception of Brugmansia suaveolens, which is thought to be from the Atlantic forest in Brazil, all the rest of the species are from Western South America. Brugmansia insignis is from the Amazonian basin, basically, and the rest of the species occur through the Andes and the Pacific coast. Some of them also arrive to the Amazon. So 2 species, Brugmansia vulcanicula and Brugmansia saguinea, are more adapted to the highlands. The other species tend to occur at lower altitudes. And something that you can see from this figure is that the higher diversity of Brugmansia is in between Ecuador and Colombia. Now, this is a map from the Plants of Gods. It shows the distribution of the use of Brugmansia and other narcotic drug plants and fungi across South America. And what I want you to see here is that Brugmansia is used mostly across all of his distribution range. It is difficult to know since when these plants were used, because as I said, there is no archaeological evidence. However, it has been hypothesized that during the colonization of America, the indigenous people became familiar with Datura and North America, And they started using Brugmansia very fast as soon as they arrived to South America, maybe 12,000 years ago. There is no proof of that, but that's the theory because its use is so pervasive and they are so important culturally for all of these cultures. And medicinally, of course. And Brugmansia is used for a lot of things by traditional medicine and indigenous people. One can divide the two main uses between the narcotic, hallucinogenic, and the more medicinal ones against infections, against pain. Usually the more hallucinogenic effects are associated to magicians, shamans, taitas, although sometimes some cultures use it as to get strength, a little bit like coca leaves, and some other cultures have even more strength, if you want, uses. For example, they give it to the dogs to help them hunting, or to the children so they grow up, if you want, or sometimes they use it so the children can help them find the gold in the rivers. Here is kind of, we search for the different groups that use Brugmansia, either present or in the past, and the kind of species that they use. And what you can see here is that a lot of groups use Brugmansia, and all species of Brugmansia are used. The kind of species that they use usually depends on where the indigenous people are. So people from the Andes and the Pacific tend to use Brugmansia sanguinea, Brugmansia canicula, the highland species, and people in lowlands and the Amazon tend to use Brugmansia insignis and Brugmansia suaveolens. Another thing that you can note here is that the use of these plants is across all the Andean part of South America and Amazonian part of South America, but there is a concentration of use in the region between Ecuador and Colombia. This could be just because the plants are more diverse and abundant there, but this is a trend that has been noted for psychotropic plants. It's the region between Colombia and Ecuador, the cultures from there have more use of narcotic plants than anywhere around the world. But really the epicenter of Brugmansia use is this place known as Sibundoy Valley. So the Sibundoy Valley is a valley, an Andean valley located 2,200 meters above the sea level. And it's a valley that is located in between the Andean region and the Amazon region. And it has a huge ethnobotanical importance for many reasons that we cannot discuss here. But what I can tell you is that it is inhabited by two groups of indigenous people, the Inca and the Kamsa. Both of them have a very prolific use of medicinal herbs, both for medicine and hallucinogenic plants, for example, yahe or ayahuasca, and also Brugmansia. So I say that this region is very important for the use of Brugmansia, and this is something that was first noted to the, if you want, Occidental world, maybe so a lot of expeditions, early expeditions went through the Sibundoy, including Humboldt, and even Jimenez de Quesada went through there. But it was Hernando Garcia Barriga that discovered the cultivars of this region. And later, Schultes, during his first trip to Colombia, went to the Sibundoy, and he was amazed by the diversity and cultural importance of Brugmansia in that region. So he went with this taita, this shaman from this region, which is called Salvador Chindoy, and he showed him the cultivars that he had there. And Schultes was amazed by the cultural importance. So this taita won't let Schultes or the children get close to the actual plant. He said that it was the most powerful plant that they have there in the Sibundoy. And also Schultes was amazed by the amazing diversity of these plants. So if you remember, my first slide has this same plant, and it says Metiscodendron mamesianum. So this is the first cultivar that Schultes So, and it's a cultivar that is so strange that Schultes thought it was a different genus. It's strange because it has these very elongated leaves, which are not common in Brugmansia, and because, among other things, the petals are not fused as in other Brugmansia. But it was not the only cultivar. They have a lot of diversity there. Some of the plants that they use, some of the Brugmansia plants that they use were clearly assigned to Brugmansia species. In this case, this Guamuco cultivar is clearly a Brugmansia sanguinea, but most of them were possibly of hybrid origin. Some of them have kind of standard, if you want, Brugmansia phenotypes, like Biangan, but many of them have very strange leaves. For example, this cultivar seems like it was eaten by caterpillars. In fact, Munchira means caterpillar, or infected by some kind of virus. This is the case also of this Kinde cultivar. More importantly, these cultivars have very different uses in, well, some of them were used for many things, but they have different uses in the culture, which implies that all this phenotypic diversity was probably also accompanied by some chemical diversity in the content of these plants, and make these plants a very good system to study the evolution of these tropane alkaloids, of medicinal compounds. However, these plants were only existed in the Sibundoy Valley, which is a very small area, and they were in, some of them were widespread, but some of them were only propagated by a few taitas, a few shamans. And they were propagated mostly vegetatively. So they are very vulnerable to extinction and disappearance. And this was already in the 70s, 60s. So that has only become worse with times. Vulnerability, in fact, it's a recurrent theme in Brugmansia. So if you look in the red list of the UCN, all Brugmansia species are listed as extinct in the wild. And this is kind of a very polemic area, but the arguments to list them as extinct in the wild is that they are mostly associated to human settlements. There are no self-sustaining wild populations. Hybrids are very common. So the species cannot maintain their identity easily because the hybrids are very common. Sterility is also very common, maybe due to hybridization. Another important argument is that there are no known animals or other agents responsible for seed dispersal. These plants have very strange fruits and they don't seem to be dispersed by anything. Mostly they are propagated vegetatively, either by humans or because of breaking of branches by rivers or something like that. And seedlings are very rarely found in nature. So what are the questions that we want to answer in my lab about the history of these plants? What are the phylogenetic relationship between species and cultivars? This is something really important. We want to know, especially how cultivars are related to species. What is the role of hybridization and selection during the evolution and domestication of Brugmansia? And how domestication impacted the genome of Brugmansia? And finally, we are interested in, and I hope you will understand better, if there are wild populations. So as it will become clear later, for our study, we need to be able to extract transcriptomes and metabolomes from plants. And for that, we need to grow the plants under controlled conditions. So in the lab, our design is that we design full field trips to go and collect plants that either have seeds or if they don't have seeds that we can propagate vegetatively in the lab. So we have designed many field trips where we try to collect every Solanaceae that we find, but we focus on some groups, and Brugmansia, of course, is one of them. So we have found Brugmansia in almost all of our field trips, and we have been able to recover all the species, not necessarily in their native distributions, but in Colombia. And more importantly, we have also designed a field trip to take the cultivars from the Sibundoy. So David Esteban Granados, which I mentioned before, is a member of my lab, which was responsible to do all this work of going and taking the cultivars in the Sibundoy. In fact, he's responsible for all the collections of Brugmansia in general. In the future, so we were hit by COVID and all our field trips were kind of stopped, but in the future we are interested in going to other places where there is a documentation of use of Brugmansia by indigenous people, or where we will find species that were difficult to sample or that we could not find before. So now I'm going to show you some of the work that we have done in the Sibundoy Valley, taking the cultivars. So in most cases, as I said before, Brugmansia is associated to you humans, but they tend to be in the garden or around houses, and it's a different thing. But when we work with cultivars, we really want to go and sample things that belong to people, and we really need to have a close interaction with the people from the Sibundoy, because these plants belong to them, and they know their uses, know their pasts, and so it's a work that involves a lot of interaction with the people from the Inga and Kamsa communities. So David went last year for a month to the Sibundoy, and he was very good at doing this job of contacting the communities and asking them to share not only their plants, but their knowledge. And it started by contacting the Taitas, which are the sorcerers, if you want, of the communities, and are the ones responsible of propagating and using most of the cultivars. And this is, for example, one of the most important Taitas of the Sibundoy, Juan Mutumbajoy. But David worked with many of them. And as you can see in these pictures, I want you to get the idea that these plants, first of all, have very weird phenotypes, but also that they are very important for these people. And as you can see, not only these plants are important for them, but plants in general are very important for the people in the Sibundoy. They have a lot of different plants in their gardens that they use for a lot of things. Look, for example, at the very weird phenotype of these cultivars in here. David also, if you want to discover, it was not a discovery, but most of the reports of Brugmansia use are associated to males because the Taitas are males. However, David saw that women have also a very important role in using and propagating these plants, and they know a lot how to use them, and it's very important also for the female part of the Indian community. Fortunately, David has also found that not only Taitas propagate these plants, but also people that are not magicians, if you want, not traditional doctors, and even the younger generations, fortunately, also some of them keep loving and propagating these plants. David also had to get in contact with the use of these plants, both for hallucinogenic and medicinal purposes. So he witnesses the preparation and the use of these plants. And well, this is so, as I guess it happened to everyone here. So the COVID strike was very hard in Colombia, and we had a very strict lockdown for a long time. So here I'm going to talk about an experiment that I was hoping to get the results by now when I accepted to give this talk, but well, we don't have the results yet, but I'm going to tell you in general how we wanted to use this experiment to answer some of our questions. And we are kind of in the middle of the experiment waiting for the results, basically. So this is kind of a pilot, because we still need to do more sampling. And so what we did was we selected individuals, we grew all the plants here in the lab, and we selected individuals from Datura, which is the outgroup, as well as all the species that we had. Brugmansia arborea is not here because we have not been able to collect that one. And we also sent some cultivars for analysis. And the idea is that we're going to do whole transcriptome sequencing. We are right now doing that for these plants, and we will use that whole transcriptome sequencing to reconstruct the phylogenetic relationships of these plants. So because some of the plants, some of the species that we are sending this time were not included in previous phylogenetic studies, we will be able to know where they fall in the phylogeny. Also, because we will be using thousands of gene trees, we might be able to understand a little bit more about incomplete lineage sorting based on gene tree discrepancies, understand about incomplete linear sorting, and more importantly, about the role of hybridization in the evolution of this group. In the context, this role of hybridization, the evolution of the group, is really important in the context of the origin and evolution of cultivars. So as I said before, most of the cultivars from the Sibundoy are thought to be Brugmansia candida, which is a hybrid between Brugmansia aurea and Brugmansia versicolor. So this is something that we hope we will be able to test with our transcriptomic data. And we might find that it's actually the case, and there was a single origin of all of these cultivars, and they are actually hybrids between Brugmansia aurea and Brugmansia versicolor. But we might find much more complex scenarios where there are multiple types of hybridization. Some cultivars belong to some species, some cultivars are hybrids, some not. And this is probably what we will find. And that will tell us about the complex history of domestication of these plants. One thing that we actually were not wanting to study, but like it came to us, it was about the existence of wild populations. And even though we didn't want to study that because it was kind of already accepted, it's really important when you study the process of domestication to understand what are the wild relatives. And as I said before, it's supposed that there are no wild populations or wild relatives of Brugmansia, that they are all associated to humans. And it's a very polemic area. But one of the arguments for that is that there are no dispersors of seeds. And some people say that they were probably big mammals that got extinct in the past. However, in our field trips, we have found fruits that are eaten by small animals, either birds or mammals. And these animals seem to be targeting the seeds. So that maybe tell us that there is a little bit more to say about that. And in this area, I really want to call to all of you Solanaceae biologists to tell us if you also have seen these things, how do you think they should be interpreted? Because, well, we will continue to do field trips, but of course, I am sure that many of you have done hundreds of field trips and can tell us if you have found this already before. Another very important thing that we found in our field trips is that we have gone to very undisturbed and remote regions, and we have actually found Brugmansia plants there. Because it is difficult to move in these places, it is difficult to know if this is a big population or not. But what we have been able to see is that these plants actually have seeds, which means that they are having a successful reproduction. That could be through selfing, we don't know, but they are producing seeds. And also importantly, you have seen seedlings, plants that have originated from seed, which means that this reproduction seems to be working efficiently in these wild populations. And again, this is very anecdotal evidence, and I would really like to hear about any evidence that any of you might have about finding these Brugmansia plants in wild locations, because we really would like to find more of these plants in the future, more of these putative wild populations. And why is that? It's very difficult. The discussion about wild or not in Brugmansia is a very deep one and a very complex one. But one thing that we can do is look at genetic diversity. It is known that the domestication of things reduces the genetic diversity through bottlenecks associated to selection of traits that are important to grow these plants in association to humans. One thing that we can do is we have plants that are medicinal cultivars, which are supposed to be under strong selective pressures by humans. And then we have these other plants, which I'm calling ornamental, which are the plants that we collect in the field nearby houses. They are not really the ornamental, and they have an ornamental purpose for these people, even if they are not the ornamental varieties that you will find, for example, on Craigslist or on, that you can buy in the United States, Australia. But they are grown for ornamental purposes. And we also have these few putative wild populations that we have found. So one thing that we can do with our data is to compare genetic diversity between these groups. And we expect that the medicinal cultivars will have a lower genetic diversity than the putative wild populations. Working with Brugmansia, it is essential to use, that's what I think, it's essential to use herbarium samples. And this is again a point where if you work in those herbariums and you find this interesting, it will be great if you can contact us. Herbarium samples are essential with Brugmansia for many reasons. One reason is that the taxonomy is quite complicated. So we really would like to be able to include the types in some of our phylogenetic analysis to help us solve some of these complexities with identifying plants. Another reason why we want to use herbarium samples is because a lot of the cultivars that we want to analyze have been lost or are very difficult to get, and they can be found in herbarium. So using these samples will allow us to understand the genomics of these cultivars that are maybe lost, or maybe understand how from the past to the present they have gained or lost diversity. This is something, of course, you cannot do transcriptomics with herbarium samples, but we can use techniques of sequence capture to be able to do genomics with these samples. Either something like 353 or a combination of things. And it's something that we really want to apply for grants to be able to do. Fortunately, a lot of the important samples for Brugmansia are in the herbarium from the university. So we can start working here. Okay, now about metabolic variation. So what brought us to my lab to Brugmansia is the compounds that they produce. I mentioned before that Tropane Alkaloids are potent medicinal compounds, and they have a lot of diversity, as you can see in this slide from the structure of alkaloids from Brugmansia. We know the uses of some of them, but we ignore the effects of many of them and how they are produced. Usually alkaloids are thought to be defensive substance. So they are used for the plants to defend against herbivores or pests. And it is something that is difficult to prove, but in the case of tropane alkaloids, it has been shown that it's toxic to some pests like Sphaerothera fructiperda, while other organisms can feed on these plants and either sequester the substances to defend themselves against predators, or excrete them or metabolize them. One interesting thing about Brugmansia is that the flowers of these plants, both the petals as well as the nectar and the pollen, contain large amounts of tropane alkaloids. However, they are visited by a lot of animals from very different groups. So that's interesting because it means that either these animals don't care about the effects of these substances or they have a way to avoid them. Another very interesting thing about the flowers and the effects of Brugmansia is that in South American people believe that if you stay close to a Brugmansia plant or you sleep below it, you might get intoxicated or even die. That would mean that there is a volatile substance that is producing these narcotic effects. This is something that has not been shown. So there has been analysis of volatiles produced by Brugmansia, but none of them seem to be toxic. And I think it might be due to pollen inhalation by people. That is something that could still be researched, and it will be great if one can find a volatile narcotic substance. So tropane alkaloids are a multi-million-dollar industry because tropane alkaloids have a lot of uses in traditional medicine, not traditional, modern medicine. And this is due mostly to the anticholinergic properties that they have. They are used mostly for motion sickness and for pains and also for treatments of the eyes, but also they are being used more and more to treat complex neurological diseases. They are so important that they are listed as part of the World Health Organization list of essential medicines. And the tropane alkaloids for the pharmacological industry are produced in many ways. Some of them use the biotechnology, as I will talk later. However, the main way to produce these compounds is through plantations of hybrids. Oh, do you hear me? I'm I'm getting that my connection is unstable. I hope you can hear me well. Duboisia hybrids. So Duboisia is also solanaceous plants, and these are plants from Australia. These hybrids are the main source of Tropane alkaloids. And the main problem of that is that these plants are grown in a very small area of northern Australia, which makes them very vulnerable to climate change and other diseases and anthropogenic associated problems. And actually, there has been some shortages of these compounds lately. For that reason, there has been a push to produce these substances through metabolic engineering. And in a huge feat of metabolic engineering, there was this paper in Nature in September of this year where they produce tropane alkaloids in yeast, and this is something that involved the inclusion of more than 20 genes and the and putting these genes in different compartments of the cell. So it's a huge work, and it's still we still don't know if it will really replace the Duboisia plantations, but something that will really help this system is, on one hand, finding genetic variants that either increase the production of some of these compounds, or genetic variants that help to produce other compounds that could have interesting medicinal properties. And this is something that we can really learn about from looking at genetic diversity in the Solanaceae, genetic and metabolic diversity in the Solanaceae. Another thing that we can do is to search for alternative sources of tropane alkaloids that can be used in plantations. So this is a list, an incomplete one probably, of the genus from the Solanaceae that can produce tropane alkaloids. And my point here is that Brugmansia is really a good system, maybe not the only one, because of his high biomass, fast growth, easy propagation. It's amenable to transformation. It's the different species are adapted to multiple environments. It's easy to hybridize. And the most important, it has a lot of genetic and metabolic diversity. And maybe one of the greatest proofs of this metabolic diversity comes from the thesis, the master thesis of a member of my lab, Sofia Rojas Contreras. She did her thesis in the Los Andes University with Santiago Madriñán. And what she did was to characterize the tropane alkaloid content of 70 plants belonging to 5 Brugmansia species, as well as many hybrids. And so I'm showing you here a summary of that. So each, if you want, column here is a different tropane alkaloid. And the color of each cell tells you how much of this alkaloid there was in average in each of these categories. And what you can see is that there's a lot of diversity of tropane alkaloids, that there is a lot of diversity on which ones each species produce. And also, a very important fact is that the cultivars, medicinal cultivars shown here, are the ones that have the highest diversity, as shown by the fact that almost no cell is gray, so they produce almost all of the tropane alkaloids, and the amount that they produce is also very high for some of them, as you can see by the dark colors there. So in a multivariate analysis of this metabolic variation, Sofia could recover a phylogenetic signal. In other words, there was a chemical identity of each species. However, these were plants collected in the field. Yeah, and so when she did an analysis by location of collection, she found that there was also a strong effect of the location. More importantly, she found that the medicinal plants from the Sibundoy were very different from the rest. This is very interesting, but because the genetic and environmental factors cannot be distinguished here, we need to grow the plants under controlled conditions to know how different, for example, the Sibundoy cultivars are from the rest, if they are really so different. Okay, so our main questions are: how plastic and tissue dependent are metabolic profiles? What is the genomic basics of metabolic variation? And what Brugmansia varieties and genes show potential for production of medicinal substances? And finally, is there evidence of selection in tropane alkaloid genes? So what we did was to bring seeds or cuttings from the field. We grow them in a growth chamber and then transplant them to a greenhouse where we have common and controlled conditions. And then what we have done is extracting metabolites from all of the plants that we sent for transcriptomic analysis. So for these plants, we will have the metabolic profiles, we will have gene sequence and expression. One thing that I put as a question, and is not really that we want to answer the question in general, but more for our work in the lab, we want to know what tissue we can use in future screenings of plants. So we are going to compare leaf tissue against full seedlings, which are two kinds of tissue that are easy to get in the lab for our analysis. Leaves are also the tissue that is mostly used for medicinal purposes by traditional medicine. Another thing that we also want to test, and is again in the context of the work that we do in the lab, not in general, we know that there is variation in the content of tropane alkaloids of different tissues, but what we want to know is how it impacts our work. So we have different ways to get our plants. One is through cuttings, the other is through seeds, and we also want to know how different those are from the plants that we collect in the field. So we have a plant that is growing in the campus of the university, and we have both taken seeds and cuttings from these plants, and we are going to compare the metabolic profiles of these three tissues. For that, we will work in close collaboration with the Max Planck Institute of Molecular Plant Physiology, who we are analyzing our samples. They will use LC-MS to do this analysis. And what we want to do is to be able to find relationship between these three things: the metabolome, the transcriptome, and the variant, so the genetic variation. We have to say that until now there is a caveat, and is that our genetic variation comes from transcriptomes, and there is differential allele expression. So that's a caveat of our variant data. But one thing that we can do is, for example, search for polymorphisms where plants with different genotypes produce different amounts of tropane alkaloids. Yeah, those are very important, interesting polymorphisms. Another thing that we can do is, using gene expression, we can search for genes whose expression cause a reduction or an increase in production of tropane alkaloids across plants. So each of these points here is a different plant, if you want, either an accession. Another analysis that we can do with gene expression is a co-expression analysis, which is to look if there are genes whose expression is correlated to other genes that we already know belong to the tropane alkaloid group. That is an analysis that can help us identify new genes that are involved in the production of tropane alkaloids. And this is something that has been done in other crops, this kind of analysis. But the problem here is that we have plants with very different degrees of genetic relatedness. So we have to correct for that. And this is something that we will do in collaboration with the Max Planck Institute, but also with Liliana Lopez-Kleine from the university. And one thing that we would like to know is if there is selection in genes of the tropane alkaloid pathway in medicinal cultivars. So for that, we can compare the transcriptomes of these plants and look for genes, or if you want, regions of the genome, where there is lower genetic diversity in medicinal plants. Those are regions that are likely object of selection, of natural selection, not natural, in this case, human co-selection. And we want to see if those genes are also tropane alkaloid genes. In other words, if there was selection of tropane alkaloid genes. And for that, we would like to use, of course, our cultivars that we use here, but also it would be great if we can use herbarium samples. The idea is to consolidate all this data to be able to integrate the phylogeny of the species with the gene expression and sequence and the metabolic profiles to understand which genes actually regulate the production of these compounds and what is the history of these genes. And finally, I hope I will have time for this section. So as I mentioned before, there is a lot of variation in leaf shape in these cultivars. And these leaf shapes, some people suggest that they look caused by a virus, by the infection of a virus. However, some evidence indicates that it has a genetic basis. On one hand, if you irradiate with UV plants from Brugmansia, you sometimes get these weird leaf shapes. On the other hand, all these cultivars have a lot of chromosomal anomalies, which might be causing these very weird phenotypes that you will never see in natural populations. And the final evidence is when normally these cultivars are sterile or are difficult to reproduce, but when you get seeds from these plants, the progeny usually shows the phenotype. So all of this seems to indicate that there is a genetic basis for that. But in the lab, we got some kind of by accident evidence that at least kind of have us thinking. On one, so David brings these plants to the lab, but he has the hobby of growing them in his house where there are more pests that might be transferring viruses or things between insect pests that might be transferring viruses between cultivars and between plants. And what he have found in his house is that, for example, some cuttings that were from a plant that did not have a strange leaf shape, started showing, for example, this culebro, this elongated kind of leaves. Another strange thing that he found is Brugmansia sanguinea, who doesn't have this strange leaf shape, was close to a plant that did have a strange leaf shape, and it kind of acquired the strange leaf shape, and it seems like it's caused from some kind of viral infection. This is interesting in the context of this study, where they found that plants of Brugmansia around the world are infected by this virus, which can cause a strange leaf shape. And the most interesting part is that this virus seems to be under selection by humans and was originary from the Sibundoy, where all these medicinal cultivars come from. So this question is too vast to really be able to answer it, at least with the experiment that we have. Now, but one thing that we can do with our transcriptomes is we can search if there are some viruses or things that are present in cultivars or in things with weird leaf shapes and not in the other species. And we can also compare this Brugmansia sanguinea plant. We have some seedlings that have normal leaves with the plants that have the curly leaves, and see if this putative virus or whatever is also present in there. We are also asking for grants to assemble the genome of some of these cultivars, and we hope that this assembly will help us know also how much how many parts of the genomes have been lost or rearranged in a way that could cause these leaf shape changes. And this is something that we will do with Born Isabel. And just to make things a little bit more interesting and complex, it is known that many of the substances that, well, it's becoming increasingly accepted that many of the substances that plants produce and that we use are actually produced in coordination with endophytes, with usually fungi that live in the plants. And this seems to be the case, at least for Daturas. It's something that we would like to know about in the future. And maybe we will be applying for grants to do it with Clarines Bermudez, who is expert in metagenomics and can help us with that and the viral project. And with that, I want to thank a lot of people, especially all of you for attending, Chelsea, Andres, and Rocio for organizing these great seminars, and all of these people that have collaborated with me. Thank you.
Okay, thank you, Federico, for this wonderful talk. Now we have a few minutes for questions. If someone has a question, please just raise your hand, just turn on your microphone or your camera, or just test in the chat box. We have some minutes for questions. Okay, I'm gonna open the chat here just to check. Okay. I had a question about Brugmansia vulcanicola. What do you think about the taxonomy between someone? Rocio. Okay.
Oh, but Stacy,
Yeah,
no, but Stacy, drop a question first.
Oh, where? It's here in the chat. I can see that.
Yes, it's in the chat.
So Stacy said that it was great to see all this research in this fascinating genus, and she was wondering how well the tropane alkaloid pathway is known. For example, can the variation in production of tropane alkaloids be related to candidate genes? Okay.
Yeah, it's relatively well known. So it's well known enough that we can produce it in yeast, which is a lot. We don't know much because I think there is no tropane alkaloids producing plant that has a complete genome. We don't know so much about the distribution of some of these genes in the genome, if they form clusters and these things. And we might not know some of the important genes that are not essential to produce the most medicinal compounds like scopolamine and atropine, but it is relatively well known. Yes, it is. And of course, we want to use a candidate gene approach to search in our transcriptome. And that's part of this co-expression analysis that I mentioned before. So what we want to do is pick some of these genes that we already know are from the pathway and see and search for other genes whose expression is correlated to these genes across varieties. But certainly there is a lot known and a lot unknown, fortunately. I think there is not so much known about how allelic variation, for example, affects the production of a compound or affects whether this compound is present and not this other one.
Thank you, Federico. Here is in the chat box is another question from Carolina Carrizo. She said that she maybe missed this part, but she's asking about which methods are you planning to use to sequence the DNA from herbarium specimen?
Well, this is one of the sections where I'm actually wanting more to hear what people think that. So what we have been negotiating with, Jesus Christ, I forgot the name now. Well, the people that do 353.
The Patol people from Q, you said.
Yeah, so we want to negotiate using 353, which I think has been very successful in herbarium samples, and put some tropane alkaloid genes into the platform. That's our idea for now. In this COCA study that I showed you before, they use a different method, and I'm not able to compare if they are actually more or less successful. I have heard in these Solanaceae talks before that the 353 is actually good for Solanum at least. So I am tempted to use that one, but that will be the thing. But we need to add tropane alkaloid genes into that because we don't only want to do phylogenies. We want to know at least the evolution of those genes. Also, we would like to put flower color genes in there too.
Nice.
Because it's such a good system. The problem is that we don't use flowers as a tissue. So because we use transcriptome, if we don't use flowers, we might not get the color gene so much. That's the only problem. But it's a very good system also to use to study flower evolution.
Very good.
Color
It's another question here from Gabriela Doria. She's asking about how long do the plants take to produce flowers? Are you getting flowers under greenhouse conditions? Also, how are people in the Sibundoy Valley propagating the cultivar mostly? Cuttings or versus seeds or?
Cuttings. So most of these things are, I won't be able to say what proportion, but most of them are sterile. You won't be able, some of them do flower in the field and even here, but it's not easy to cross them and you don't usually get seeds. They are propagated vegetatively, which is super easy. Is good and bad for us. It's good for us because we have genetically identical plants, which is good to do genetics. It's also good because we get the plants to flower very fast here. So if we grow them from seeds, it will take much longer for them to flower. When you bring cuttings, they just flower very fast. The problem is that we bring a little bit the environment with them. In terms, for example, of plagues and things, because it's a cutting from a plant that was outside, we cannot completely delete the environment that the plant was before in terms of epigenetics. But it's great that they flower very fast here. It's not easy to do genetics, classical genetics with these plants. On one hand, they are not easy to cross, and on the other hand, well, they can be easy to cross, some of them are. But the other problem is that, of course, generation time is long. So we can get the F1 easily, but then to get the F2, it's a little bit more complicated.
Thank you, Federico. We have here another question from Clarines Orozco. She's asking about the relationships, phylogenetic relationships that you show could be affected when you use a broader sampling. Maybe you include the Vulcanicola or the another species I didn't remember, or maybe one of these cultivars. You think that phylogenetic relations are stable, or they can change in the future?
Okay, so the phylogenetic results that I show are hypothetical. No? We don't have anyone yet. But it's based on the Dupin and Smith phylogeny. And I'm assuming that the other species are the Vulcanicola and the Insignis are going to fall in those two clades because we know they are from those two clades. But it's hypothetical. I hope that the inclusion of the cultivars won't change relationships, and actually it's kind of interesting and difficult to predict how some things that are hybrids and potentially hybrids between multiple species will fall within a phylogeny. But right now we don't really know. This pilot experiment that I showed, we are only, at least for transcriptome, for metabolomics, we are sending multiple individuals per item, but for transcriptomes, it's just a pilot and we are sending just one individual. So we won't know, for example, if each species is actually monophyletic. That we won't know at this point, but it's something that we will do in the future once we have more species, more individuals from each species. And I think what we will do is first the phylogeny only with species and then try to put the cultivars in there. But even the clear species are very difficult to, it's very difficult to assign items to species on one hand. A lot of them might be hybrids, and just I am a person that don't like to call hybrids by the phenotype. I think that's a very tricky area.
And especially Brugmansia.
My cartoon is very nice because it's easy to understand it, but we might get things that are very complicated. And I expect to get things that are very complicated. Maybe some of the species might not be monophyletic, a lot of hybridization. We do know that it happens a lot. So I really don't know exactly what to expect.
Okay, thank you, Federico. We had another question here from Aureliano Bombarelli. He said that was a very interesting talk, and he has two questions. Do you know if there is polyploids in Brugmansia, including the mostly cultivated ones? And another question about the aberrant leaves. It's like virus. If you don't find virus, have you considered active transposon affecting leaf changings?
Yes. Okay. So the first question
about polyploids.
So these things kind of, the diagnosis change with the technique that you use. But for the cultivars, there are all the karyotypic studies, which I think I showed you before, and they don't show polyploidy, but they show aberrations in the number of chromosomes. That yes. However, I'm not experted in cytogenetics to know if the techniques in the 60s were good enough for today's standards. So there might be polyploidy. We actually don't know anything about these cultivars from the genetic standpoint, except from the cytogenetics. And the other transposons looks a lot like a transposon or virus thing. Yes, we have this very circumstantial evidence that seems like the thing can be transmitted between plants, but it's something that really needs to be tested. But if it's genetic, I would say that it's probably either a transposon, a virus, which might have similar effects in the genome, or just chromosomal loss of chromosomal regions and things like that. And yeah, I don't know if any of you know about any other plant that has this kind of leaf shape, and if that is not caused by a virus.
We have so many questions today. Here is another question from Rocio. She said that had an unstable connection, and she leaves her question here. Do you have an hypothesis about the potential fruit seed dispersers for Brugmansia? These fruits eaten by some animal there.
Look, this plant, one thing that doesn't fit exactly the story about wild or not wild, which is a very complicated one, is that these things were found relatively close to human settlements. Could be a rat. It could be anything. I don't know about the thinking of, by looking at the things, I would say either a rat or a rodent, something like that, or a bird like a parrot or something like that that has a kind of piercing beak, but that's very sloppy kind of intuition from me, yeah. But it was not like in super wide places that we found these eaten fruits. So it might not actually be eaten by a rat, by a stoned rat or something like that, and doesn't have any effect in the dispersal of the plant. But it's something that we want to keep investigating, and hopefully if any of you have found any of these things, can send us the evidence or the photos.
Thank you, Federico. We have another question here from Gustavo Silva. He's asking about how much domesticated or in which state of domestication are expected to be Brugmansia species in the natural range of the genera. You think could be really some of these species are more domesticated than others, or some that are wilder or something?
Yeah, so Brugmansia has many things that make it fascinating and difficult to study in terms of domestication. It's fascinating because, at least to my knowledge, there are not so many evidence of cultivars and variation associated to medicinal use in Brugmansia or in other plants. So it's great. And we don't know so much. We know about crops. Generally, you have selection on the same traits. Plants need to grow fast. They need, if they produce grains, they need to hold their grains. So it's usually the same genes. It is not well known for medicinal plants what that will be. Another difficulty about, so we don't know what to expect. Another difficulty with Brugmansia is Brugmansia, there is no wild thing, at least according to what is accepted by people. So we have not actually anything to compare it, a really wild thing, except in the case we can prove that these things that we find sometimes in small jungles are really wild things. We don't know what a wild Brugmansia actually is. We expect these cultivars to have lower diversity, and phenotypically they are so different that we expect big changes, but we don't know what to expect from the domestication literature. There is really not much about medicinal plants yet.
Okay. We had a comment here from Carolina Carrizo that say that the people in Cotiana are using also that. I think she's talking about the library from Pato, the 353 genes. I think it's a comment about that. Gustavo said that was a great talk. Thank you.
Thank you.
Clara Ines is asking about what about the cultivar plate? Is well differentiated from other plates, or not yet?
No idea. No idea.
Gina Paola Sierra said that was a great talk. Have you ever found a cultivar or a species which cannot be propagated by seeds?
Yeah, I think that David is better entitled to answer that, but I think she means that they do produce seeds and the seeds don't grow. I wouldn't know. A lot of them don't produce seeds. Now, we have not tried to cross them. We just have been in the field, and if they have seeds in the tree, well, we collect them, but we have not actually crossed them or anything, because we will lose the identity of the cultivars. But I am not entitled to say once they produce seeds, if the seeds tend not to be viable. I would tend to think that it's probably the case, but I don't know the answer. Yeah, David probably knows.
You have to ask David. Okay, thank you, Federico. People here here is another question. Well, Clarence is just trying to clarify that there is a phylogenetic tree. That is the tree from Dupi.
Oh, yeah.
Okay, Federico. I think this is all questions. Thank you so much, Federico, for this wonderful talk. It was really amazing. And for next week, we're gonna have to Joseph Jacobs from the University of California at Davis. That he's gonna tell us about the breeding, plant breeding and germ plants improvement, which focus in Solanaceae. That is all for today. Thank you so much. Happy Thanksgiving to the people following us from US, and see you next week. Enjoy your weekend. Thank you. Thank you so much.